Okay, let's continue on telomeres. So these are the ends of the chromosomes. So during each cell division, when DNA replicates itself, it creates a problem with the lagging strand. So this is how it works. Okay, so this is what the issue is. Now remember, the lagging strand goes from 5 to 3, right? So when you make the new DNA, you can't go 5 to 3. Because in this case, the two strands are not antiparallel, right? So what you do is you have to synthesize the strand from 5 to 3, but you're going to do it the other direction. So if the template strand is 5 here, that means this is going to be the 3 for the synthesized strand. So the polymerase is going to jump over here and then extend the strand from 5 to 3, right? So again, it's going to come here and do 5 to 3. Don't worry about this leading strand. It's just going to make things confusing because they're trying to show both, but don't worry about that. So let's say all these are Okazaki fragments, right? Because again, the polymerase jumps ahead and then they go backwards to synthesize the new strand from 5 to 3, right? So this is 5 to 3, 5 to 3, 5 to 3. Okay, so this is the lagging strand. And this is the new DNA strand that's complementary to the lagging strand. And then these are called Okazaki fragments. Now again, for each Okazaki fragment, or for any continuous new DNA strand, you have to have a primer first, right? You gotta have this primer. Otherwise, DNA polymerase cannot work from nowhere. It has to have a little anchor, a little fragment where it can, extend. So at the end, the primers will be taken out, right? So it will be taken out because it's RNA, and we can't have RNA in a DNA strand. So the RNA primer fragments are taken out, and they will be replaced with the DNA by DNA polymerase. So the DNA polymerase, again, will just kind of synthesize a short fragment, right, of DNA, and that will replace the previous RNA primer. Now, it works for the primers in the middle of the strand, but when you get to the last Okazaki fragment, when you get to the end of the chromosome, after this primer fragment or segment is removed, the DNA polymerase cannot synthesize a DNA segment because there's nothing here for it to kind of anchor to. There's nothing here for it to extend the strand, right? There's nothing there. So that means after each cell division, you are going to lose a short segment of the DNA, right? Or you're going to lose a short segment of chromosomes. Chromosome is partly DNA. So every time cell division, if this is the original strand of the DNA, so every time after each cell division, the strand will get shorter and shorter and shorter. And this has been shown to be related to aging. So this is a very bad thing, and we don't want that to happen. So our body has a mechanism. We produce an enzyme called the telomerase to prevent the shortening of the chromosome. So basically, this picture shows you how it works. So basically, it will kind of create a short segment for the DNA polymerase to anchor on, and then the DNA polymerase can go from 5 to 3 to make a short DNA segment to replace the primer. So this way, you don't lose a short segment each time cell divides. So this is how it works. So basically, it will provide a little segment here for the DNA polymerase to grab onto, to extend, and replace that RNA primer with DNA. Okay, so if you have very strong telomerase activity, then your chromosomes won't, shorten. So you probably will age a lot slower than other people. We're going to mention DNA mutation real quick. we'll have more details regarding the specific types of DNA mutations when we talk about gene expression. Alright, so here just a quick overview on DNA mutation. So what is a mutation? A change that occurs in DNA sequence. Right? So let's say this is the original sequence. So you have TAACT. Now there's a mutation, so now you have TAACC. So this one single nucleotide change may not affect anything. And in most cases, it really doesn't, because we have some mechanisms to kind of buffer this kind of mutation. But bad things do happen. So sometimes a one single nucleotide change can affect what amino acid you have, and this could lead to the change of the entire protein function. So a lot of the genetic disorders are kind of related to DNA mutations because you're making a dysfunctional protein product. But mutations are still good because this is the source for speciation, right? There are so many different species living on this planet. We all have different characteristics. We have different genetic makeup that kind of make those different features, different characteristics. And that's all thanks to mutations. All right, now when we talk about classification of gene mutations, we're going to kind of classify them into two major groups. You can just skip this slide. Like I said, we'll talk about this in more details later. Okay, so for the two main classifications, we have hereditary mutations. So based on the name, that you can kind of guess that these type of mutations can be passed on to offspring. Okay, so these mutations are inherited from a parent, and the mutation is going to be present throughout the person's life and virtually in every cell in the body. Okay? So what's the source for the hereditary mutations? Now, when an egg and sperm cell unite, if one of them is carrying mutations from a parent, then the resulting fertilized cell, which is called a zygote, will have this mutation. And then zygote, so everybody really comes from a zygote. So this is the resulting cell from fertilization called the zygote. So the zygote will divide into two cells, and those two cells will further divide. So now you have 4 cells. And these cells keep dividing. So you have 16, 32, 64, 128, and eventually all the cells in your body. So if the zygote has the mutation, remember we're doing DNA replication, right? So all the following cells, all these following cells will have this mutation. So the baby or this new person will have the mutation in virtually every cell in the body. And this person inherits the mutation from the parents. So this is called hereditary mutations. The second type is called acquired mutations or somatic mutations. So you acquire this mutation at some point in your life. It's not from your parents. It just happens at some point in your life. So there are different sources. The mutations can be caused by environmental factors. We just talked about UV radiation earlier, right? And there's also some chemicals can also cause mutations. Now, a second source is we talk about some mechanisms to kind of correct the errors in DNA, right? But once in a while there will be mistakes that skip the surveillance system. So you may have an error that's retained in the DNA. So a mistake is made as a DNA copy is dissolved during cell division, and this mutation somehow is not detected and is preserved in the DNA. And then when that cell makes new cells, when that cell divides, then all the following cells will have that mutation. So this is acquired or somatic mutations. Now these mutations happen in somatic cells, all the cells except sperm cells or egg cells. So muscle cells, skin cells, hair cells, so those are all somatic cells. Now acquired mutations cannot be passed on to the offspring, right? Because your muscle cells, your hair cells, your skin cells do not contribute to the genetic makeup of the baby, right? So only the DNA in sperm cell or egg cell will be passed on to the baby. So that's the two types of mutations. Now this is a very interesting term. mosaicism. So somatic mutations that happen in a single cell early in embryonic development can lead to this situation. So this is how it works. Remember we said after the egg and sperm cell unites, you're going to have a zygote. Right? And then the zygote at the initial stage of embryonic development, it's going to divide it really fast. It's going to make 2 cells, and those 2 cells divide, so now you have 4. And again, these 4 will divide again, so you double that, that's 8, right? So I'm just going to draw 8 cells. So let's say in this stage, there is a mutation. So let's say this cell has a mutation. So this cell is going to develop into more cells, right? Okay, along with these cells. So only the cells that originate from this cell will carry the mutation, right? So the cells that come from these cells, they have the normal DNA sequence. They do not have the mutation. So this creates a situation where only certain cells in your body have the mutation. So some of the cells are normal, some of the cells are not normal. Okay, so this is what this term is about. So there you go. All right. Polymorphism. So genetic alterations that occur in more than 1% of the population are called polymorphisms. So just think of it as the fact that we all have different DNA sequences. So our genes, even though it's the same type of gene, we have different versions. So polymorphisms are responsible for many of the things, many of the different features that you see between people, right? We have different eye colors, hair colors, we have different blood types, and some people have, let me give you this example. Some people may be able to drink more alcohol than others. They don't get drunk very easily. That's also polymorphism. It's really kind of determined by what version of genes you carry. And then another example is response to different drugs, different medications, right? So some people may have a very strong reaction, while certain people are very tolerant of the drug effect. So that's also kind of polymorphism. So when they develop drugs, they need to kind of take into consideration of how people may respond to the drug differently. And so that's polymorphism. And that's pretty much the end of the lecture. So let me stop recording.